Welcome back to the Deep Dive. This week we're diving into a new batch of papers from longevitypapers.com.
Yeah, particularly the ones published today, February 27th, 2025.
We're looking for those aha moments, those findings that make you rethink what you thought you knew about aging.
Exactly, and we're really zeroing in on the research coming out of those top institutions, the ones we all know and respect.
Right, like that aging cell paper, the one using circular tRNA to treat cells affected by HTPS.
Oh yeah, that one's a game changer.
Longer telomeres were almost a given, but to see reductions in senescence markers, inflammation, and even DNA damage.
It's remarkable.
And they even saw a normalization of nuclear morphology.
It really does challenge the way we think about tackling telomere dysfunction.
Yeah.
We've been so focused on linear TERT mRNA, and now suddenly we have this circular RNA showing such potent effects.
Absolutely. And for those of us working on delivery
It
mechanisms,
opens up so many possibilities.
imagine the potential if we can crack the code on stability and effective delivery of circular RNA for in vivo applications.
It could be huge. And while we're on the topic of game changers, we have to talk about that Nature Communications paper on senescent cell identification.
Oh yeah, FINEPI.
What a name, right?
I know. But an algorithm that defines cell-type specific senescence signatures.
That's groundbreaking.
We've been struggling with these generic markers for so long, trying to pinpoint senescent cells in different tissues. This could be the precision tool we've been waiting for.
Imagine being able to see exactly what a senescent skin cell looks like compared to, say, a senescent neuron.
The possibilities for targeted therapies are endless.
Absolutely. And then there's that paper published just today in Hematopoietic Stem Cells on ZSCan4.
The one exploring its potential for treating those ultra-short telomeres in dyskeratosis congenita patients.
That's the one. We've always been fascinated by ZSCan4's ability to elongate telomeres via recombination, but to apply that to such a specific patient population is really pushing the boundaries.
It's a delicate balance, for sure.
Yeah.
The potential benefits are huge, but we have to proceed carefully.
Agreed. Okay, shifting gears a bit, let's look at some of the papers highlighting how aging is more than just the sum of its parts. It's about the interconnectedness of systems, and this week's papers have some really thought-provoking examples.
The Nature Review's endocrinology paper on the crosstalk between bone and muscle really drove that point home. Endocrine, inflammatory, nutritional, even biomechanical signals impacting both tissues.
It really is a complete reframing of how we approach interventions.
Imagine the possibilities for synergistic therapies. Could we target bone health to improve muscle function or vice versa?
It's an exciting new dimension in our research.
Yeah.
And then you have that paper from bioRxiv, the one challenging the simplistic view of pregnancy-accelerating aging.
Yeah, the one where they analyzed 62 lab tests across 300,000 pregnancies.
Exactly. And to find that some systems actually show signs of rejuvenation,
Like renal iron and liver function.
while others show signs of accelerated aging, like coagulation and thyroid function.
It really throws a wrench in those old assumptions about pregnancy and aging. It paints a much more complex and nuanced picture.
For sure. And it makes you wonder if we could potentially harness those beneficial pregnancy-related changes for longevity interventions.
Isolate those specific pathways.
Now that would be incredible. Okay, ready for another wild one?
Always.
It's about the gut-ovary axis, that bioRxiv paper where they transplanted gut microbiota from young to old mice, and it actually led to transcriptional remodeling in the ovaries.
Reducing inflammation and improving function just by manipulating the gut microbiome.
It really makes you think how much of ovarian aging is actually driven by gut health.
And could we manipulate that to extend reproductive lifespan?
It's a fascinating question. Okay, ready to dive into the brain?
You bet. Always ready for some brain-bending research.
There's a lot of it out there, but this week is particularly interesting. We're seeing research move beyond the traditional markers of brain aging and really delve into those complex mechanisms underlying cognitive decline.
Like that bioRxiv paper on the extracellular matrix, ECM, and its role in brain aging.
Not exactly a dinner table conversation topic, right?
I know. But turns out the ECM is intimately linked to both synapse health and microglia activity.
And they found these regional differences in ECM remodeling. It could be the key to developing targeted therapies for specific brain regions.
It's a goldmine for future research. And while we're on the topic of exploring new frontiers, that paper on brain imaging during sleep.
Also from bioRxiv.
Yeah, that one really caught my attention.
It's pretty unsettling to think about older adults having reduced cerebrospinal fluid flow during sleep, potentially hindering waste removal.
It really highlights the importance of sleep quality as we age.
Absolutely. And it's not just about how much we sleep, but what's happening during those crucial hours when our brains are supposed to be clearing out waste.
Makes you think twice about pulling those all-nighters.
Yeah, no more. I'll sleep when I'm dead.
Exactly. Speaking of important things that decline with age, let's talk about that Nature Aging paper on osteocalcin and cognitive resilience.
Oh, yeah, that's a fascinating one. It's amazing how a hormone produced by bones can have such a profound impact on neuronal health.
Right. But here's the catch. Those neurons need healthy primary cilia and functional autophagy for osteocalcin to work its magic.
And both of those decline with age.
Talk about interconnectedness.
It's a perfect example of why we need a more holistic approach to longevity research. Targeting individual pathways is important, but we also need to understand how these systems interact and influence each other as we age.
This is incredible, and we're just scratching the surface.
There's so much more to unpack in this week's batch of papers.
We'll be back soon to dive into even more groundbreaking research.
Stay tuned for part 2 of our deep dive. Welcome back to the deep dive.
I'm still thinking about that last paper on osteocalcin and cognitive resilience.
Me too. It really highlights just how interconnected our bodies are.
It does. And, we've known for a while that bone health is important, but to see this potential impact on cognitive function,
It's mind-blowing.
it really is. It makes you realize we need to think outside the box when it comes to longevity research.
Absolutely.
Right.
We can't just focus on individual organs or systems anymore.
It's all connected.
Exactly, which reminds me of this paper I came across that really challenged my assumptions about cancer risk and immunosenescence. It's from bioRxiv, and it delves into T-cell receptor diversity.
So how well our immune systems can recognize and fight off threats.
Exactly. They analyze a massive data set of TCR repertoires from people aged 20 to 80.
And I'm guessing they found a decline in diversity over time?
Unsurprisingly, yes. But what's really interesting is the difference they found between men and women.
Oh, tell me more.
Women experienced this decline a full 11 years later than men.
Wow. That's a huge difference.
It is, and it could be connected to the lower cancer incidence we see in women.
That's fascinating. So essentially, women's immune systems are buying them more time.
That's what the researchers suggest. They propose that cancer risk increases with age due to both the accumulation of mutations and the weakening of our immune surveillance.
It's because of that declining TCR diversity.
Right. It's like a two-pronged attack, and it makes you wonder if we could develop immunotherapies that specifically target those age-related changes in TCR diversity.
Could we delay that decline and reduce cancer risk for everyone?
That's the hope. It would be incredible to boost the immune system's ability to recognize and eliminate those precancerous cells before they even have a chance to develop.
Now, that would be a game changer. Okay, let's switch gears for a second and head back to cellular senescence. Remember that gut-ovary axis paper we discussed earlier?
Yeah, the one with the microbiota transplants?
Well, there's another paper. This one takes a closer look at how senescence affects the gut barrier itself.
Ooh, this one used a really cool gut-on-a-chip model.
I saw that. It's pretty amazing technology.
It allows researchers to study age-related changes in the intestinal barrier in a really controlled environment, and their findings were pretty significant. They found that senescence in gut cells contributes to increased permeability and inflammation.
Which is not what we want as we age.
Definitely not. It's like a leaky faucet in your gut, allowing all sorts of things to seep into your bloodstream.
So what can we do about it?
Well, this research is really a call to action. We need to develop therapies that can protect or even restore gut barrier function.
Like a drug that could strengthen those tight junctions and prevent that leaky gut scenario.
Exactly. And we already know the gut microbiome plays a role here.
Right, so maybe a multi-pronged approach, targeting senescent cells in the gut, promoting a healthy microbiome, and maybe even developing drugs that strengthen the gut barrier itself.
You're thinking like a true longevity researcher. It's about tackling the problem from all angles.
Okay.
Okay, are you ready to dive into the world of extreme longevity?
Always. Do you have a paper about centenarians?
Even better, this one's about supercentenarians.
No way.
Yeah, this paper from bioRxiv delves into the multi-omics landscape of a woman who lived to be 117 years old.
Wow. They must have analyzed everything.
They did genomic, transcriptomic, metabolomic, the whole works.
So what did they learn? Did she have some kind of genetic superpower that protected her from age-related diseases?
Surprisingly, no. She actually had quite a few health issues throughout her life. Yeah, what the researchers suggest is that extreme longevity might not be about avoiding age-related decline altogether. It might be more about the ability to manage that damage and maintain functional resilience.
So it's not about being superhuman, but about having a really robust and adaptable system.
Exactly, which is actually quite encouraging. It means we don't necessarily need to find that single magic bullet to extend lifespan.
We can focus on strengthening our systems and promoting overall resilience.
Exactly. And this multi-omics analysis gives us some valuable clues about which biological pathways and mechanisms might contribute to that resilience. Speaking of resilience, what about hematopoietic stem cells?
Ah, yes, the building blocks of our blood and immune systems.
This paper from bioRxiv challenges our understanding of how those stem cells differentiate into specific blood cell lineages.
You mean like red blood cells, white blood cells, and platelets?
Exactly. We've always thought of that differentiation process as a one-way street, but this research suggests that some hematopoietic stem cells can actually undergo bidirectional fate transitions.
So they can switch back and forth between lineages.
It seems so. And they found a key player in this process, the hex gene. It seems to regulate the balance between myeloid and lymphoid cells.
And its expression declines with age, right?
Right, which could explain that shift towards myeloid dominance we see in older immune systems.
Which makes us more susceptible to infections.
Exactly. So if we could figure out how to maintain or even boost hex expression, we could potentially rebalance the immune system and restore its youthful vigor.
Now that would be something. This has been an incredible deep dive so far.
We've barely scratched the surface.
I know, there's still so much to explore.
We'll be back soon to dive into even more groundbreaking research.
Stay tuned for part 3 of our deep dive. Welcome back to the final part of our deep dive. We've covered some truly fascinating research so far.
It's been a wild ride through the world of longevity research, and we're not done yet.
You know, I'm really glad we're circling back to that paper on age-related changes in cerebrospinal fluid flow during sleep, the one where they imaged people's brains while they were sleeping.
It was a technical feat for sure, but the insights gained are invaluable.
It's incredible technology. And to think that older adults experience reduced CSF flow, potentially hindering waste removal.
It really does highlight the importance of sleep quality as we age.
It's not just about getting enough sleep. It's about making sure our brains can actually do their job while we're sleeping, clearing out that waste.
Exactly. And the fact that this reduced CSF flow is linked to both neural and vascular changes shows just how complex aging really is.
It's a multifactorial process for sure. Even sleep is affected.
Yeah.
Speaking of complex interactions, let's talk about that paper from NPJ Aging, the one on therapy-induced senescence, calcium flux, and the mitochondria ER contact site.
Or CS for short.
Right, MERCS. It's a mouthful. But the implications are huge. We're talking about therapy-induced senescence, or ptysis, which is essentially a state of cell cycle arrest, often triggered by cancer therapies.
Right, it's a bit of a double-edged sword.
So we're stopping the cancer, but potentially accelerating aging in other tissues.
That's the concern. And this paper sheds some light on how that happens. It seems that TIS disrupts the balance of calcium signaling between the mitochondria and the endoplasmic reticulum.
Two key organelles involved in energy production and stress response.
Exactly. And this disruption leads to an accumulation of calcium in the mitochondria and a decrease in the ER.
Which impairs mitochondrial function and makes cells more vulnerable to stress.
Right. So how do we fix this? It's a tough situation. Are we stuck between a rock and a hard place with cancer treatment and aging?
That's the question. This research really emphasizes the need for a more nuanced approach to cancer therapies, one that considers not just killing cancer cells, but also the long-term impact on overall health and lifespan.
We need to find ways to mitigate those negative effects on aging, maybe by targeting those specific calcium signaling pathways.
This is why longevity research is so important. It's not just about living longer. It's about living healthier for longer. And, you know, speaking of the bigger picture, let's talk about that paper that highlights the social determinants of aging, the one from Madraxov on epigenetic age acceleration.
Ah, yes. The one looking at social disparities and health outcomes in the Women's Health Initiative study.
That's the one. It's a powerful reminder that aging isn't just about biology. It's about the environment we live in, too.
Absolutely. This study found a strong correlation between socioeconomic status and epigenetic age acceleration.
Which isn't surprising. People living in poverty often face greater health challenges and experience accelerated aging.
Right. But what's striking is that Black and Hispanic women experienced a faster pace of aging even after accounting for socioeconomic factors.
It points to those systemic inequities that contribute to health disparities and accelerated aging in marginalized communities.
We can't just focus on biological interventions. We need to address those root causes and create a more equitable society.
Where everyone has a chance to age healthily.
Yeah.
All right, let's end on a positive note.
Right.
What about that paper on the impact of aerobic exercise on brain age?
The one from a Drexel that looked at brain predicted age difference or brain PA'd.
That's the one. Did they find the fountain of youth in the gym?
Well, not quite. But they did find that moderate to vigorous aerobic exercise for 12 months led to a reduction in brain P.
So the exercise group's brains aged more slowly than the control group's brains.
Exactly. It's more evidence that exercise is one of the best things we can do for healthy aging.
And it's not just about physical health, it's about cognitive health too. Speaking of which, let's wrap up with that paper on liver aging, the one from Copper that focuses on copper dysregulation.
Copper is essential, but too much of it can be toxic. And this study found that labile copper levels increase with age in the liver.
And that's a problem because
It interferes with the activity of a protective enzyme called ALDH1A1.
so the excess copper basically shuts down this protective mechanism, potentially contributing to age-related liver damage.
It's all about balance. We need copper, but not too much. And the researchers were actually able to restore copper homeostasis and preserve ALDH1A1 activity in aged mice using a copper chelator.
So there's hope for potential therapies targeting age-related liver damage.
Absolutely. Well, folks, that brings us to the end of our deep dive. We've explored the many facets of aging, from the cellular level to the societal level and everything in between.
It's been an incredible journey through the latest advancements in longevity research.
And it's clear that we're making remarkable progress in unraveling the mysteries of aging.
It's a truly exciting time to be involved in this field.
It is indeed. So, what are your biggest takeaways from this deep dive? What will you be thinking about as you continue your research? What new questions will you ask?
For me, it's the sheer breadth and depth of research being done in this field. From manipulating telomeres to understanding the social determinants of aging, the possibilities are endless.
And as we continue to push the boundaries of scientific understanding, we're sure to uncover even more groundbreaking discoveries that will transform the way we age.
It's an incredible time to be alive and working in longevity research. Thanks for joining us on this deep dive. We'll see you next week with a fresh batch of papers and more exciting discussions.
Until then, keep diving deep into the world of longevity research.